Check. Good evening, and welcome to the 2021 Nora and Edward Ryerson Lecture. The Ryerson Lectures are named in honor of Nora and Edward Ryerson. Edward L. Ryerson, Jr. was a long-standing member of our Board of Trustees from 1923 to 1971, and served as chair of the Board of Trustees from 1953 to 1956. The Ryerson family, now the Ranney family, has an association with the university that dates back to our earlier days, when Martin A. Ryerson served as a member of our first Board of Trustees and presented the gift of the Ryerson Physical Laboratory, named in honor of his father. I'm pleased to see that we have some members of the Ranney family with us this evening, and I would hope you'll all join me in extending them a special welcome. The annual Ryerson Lecture is a very special one for the University of Chicago community. Each year, the speaker is chosen by a faculty committee, which recommends a faculty colleague, an individual whose research contributions are judged to be of lasting significance, and who the committee believes is a faculty member that the community would like to hear from and will benefit from hearing, and whose work represents in their field the highest aspirations of the university and its faculty for research of depth, originality, and impact. This opportunity to hear from an esteemed member of our faculty, chosen by the faculty, has been a central event in the intellectual life of the university since John Hope Franklin delivered the inaugural Ryerson Lecture in 1974. The collected Ryerson Lectures represent 48 years of the university's scholarly leadership across the disciplines, and also encapsulate an intellectual history of our broader society over that period. Today is the first Ryerson Lecture to be held in Friedman Hall at the David M. Rubenstein Forum, which is a beautiful new hub for the university's intellectual activity. This is also our first Ryerson Lecture since 2019 due to the circumstances of the COVID-19 pandemic. While much of our audience is attending virtually tonight, I'm pleased that we are also able to welcome a limited in-person audience for this important event. This year, we are delighted to have Neil Shubin as our speaker. Neil is the Robert R. Bensley Distinguished Service Professor in the Department of Organismal Biology and Anatomy and the college. He is Special Advisor to the Provost for Life Sciences and Associate Dean for Academic Strategy in the Biological Sciences Division. A paleontologist and evolutionary biologist whose research examines the evolution and development of vertebrate animals, Neil's research integrates diverse temporal, phylogenetic, and structural scales in order to advance the field of evolutionary biology. His work seeks to understand the mechanisms behind the evolutionary origin of new anatomical features and faunas, with a particular focus on new anatomical designs that emerged as a result of ecological and evolutionary shifts between the Devonian and Triassic periods, resulting in the transition from water to land by fish. Over the past 15 years, he has led expeditionary research programs in Canada, Africa, the continental United States, Asia, Greenland, and Antarctica, which have led to new insights on the origins of major groups of vertebrates. He is particularly well known for his 2004 discovery of Tiktaalik, a 375 million year old fossil found in Arctic Canada that has been described as the missing link between fish and tetrapods. Neil has been a major force between the unification of molecular and paleontological approaches to the understanding of evolution. Almost uniquely, he has run a laboratory that links analyses of development and genetics with those of expeditionary paleontology to reveal how changes in the activity of genes may have been associated with major evolutionary changes. Neil is a celebrated science writer who has received numerous awards for his efforts to make science communication more accessible to the public, including the American Association of Arts and Sciences Kavli Science Journalism Award and the Friend of Darwin Award from the National Science Center, both in 2014. He was honored by the National Academies of Sciences, Engineering, and Medicine in 2015 for excellence in communicating science to the general public through his Emmy Award-winning television series, Your Inner Fish, which aired on PBS that year. He is the author of 3 popular science books, the best-selling Your Inner Fish from 2008, on which the television series was based, The Universe Within, The Deep History of the Human Body from 2013, and his most recent book, Some Assembly Required, Decoding 4 billion years of life from ancient fossils to DNA, published in 2020. Neil joined the University of Chicago in 2000 as professor and chair of the Department of Organismal Biology and Anatomy. During his time at the university, he has held a number of positions, including Associate Assistant Dean for Basic Science Education in the Pritzker School of Medicine, Provost of the Field Museum of Natural History, and Interim Co-Director of the Marine Biological Laboratory following its affiliation with the University of Chicago. Prior to joining the university, Neil was a member of the faculty at the University of Pennsylvania and a postdoctoral fellow at the University of California, Berkeley. He received his AB from Columbia University in 1982 and a PhD in organismic and evolutionary biology from Harvard University in 1987. He is a fellow of the National Academy of Sciences, the American Academy of Arts and Sciences, the American Association for the Advancement of Science, the American Philosophical Society, and the John Simon Guggenheim Memorial Foundation. Neil's lecture today is titled, Finding Your Inner Fish, Fossils, Genes, and the History of Life. Before I hand things over to Neil, I'd like to take a moment to acknowledge the past Ryerson lecturers who are watching the virtual presentation tonight. The Ryerson Lecture is an important event in the University of Chicago's academic calendar. It highlights the remarkable intellectual contributions of our faculty to the broader academic community and continues to enshrine rigorous inquiry as one of the university's highest ideals. These scholars come from across the university's schools and divisions and represent a diverse array of intellectual achievements, embodying the ideals that continue to define the University of Chicago. Before we begin, I'd like to acknowledge the passing of 3 of our Ryerson lecturers since we last gathered for this event in 2019. 1992 Ryerson lecturer, Marshall D. Solons, who passed away earlier this month. 1981 Ryerson lecturer, James M. Gustafson, who passed away in January. and 2019 Ryerson lecturer Michael Silverstein, who passed away in July 2020. As valued scholars, colleagues, and friends, their loss is deeply felt by our community, and I would like to extend my condolences to their families. Now, please join me in welcoming Neil Shubin.
Good evening. Thank you, President Zimmer, for the very generous introduction. And thank you, my faculty colleagues, for giving me the distinct honor of being the 2021 Ryerson Lecturer. It's, to be recognized this way by one's colleagues is a distinct privilege and honor. Well, I also have the privilege tonight to talk to you about history. We're going to be talking about history at sort of three different scales. We're going to be talking about history at the scale of hundreds of millions of years. Great transitions in the history of life happens over those time scales. We're going to be talking about history in terms of development and growth, from egg to adult, the development of bodies. That's important for understanding evolution as well. And finally, we're going to talk about another form of history, which is my own life. Not from egg to adult, of course, but in another slice of time. So I hope you indulge me if I begin with the last one first, to talk about how I got into this field, because it has a distinctly University of Chicago origin. I was a first-year graduate student trying to figure out what I wanted to do for a PhD thesis. And I was taking a course on the greatest events in the history of life. And each week was a different great event in the history of life. And it was almost like speed dating with some of the great moments in Earth's history and life's history. And in one of the first weeks, the professor showed this slide. This slide was done by my predecessor, I didn't know it at the time, who was chair of the anatomy department at the University of Chicago, Len Radinsky. And what he did in this diagram was to distill the transition from life in water to life on land to a very simple cartoon. Really, this was an introductory textbook. And what you see on top is a fish. It's actually a cartoon of a fossil fish, a lobe-fin fish. And on the bottom is a cartoon of a fossil limbed animal, one of the first critters to walk on land, a fossil. I remember looking at this slide and thinking, that is a first-class scientific problem. Trying to figure out how animals evolved to walk on land. Virtually everything has to change. Anatomical organs have to arise. There's new genetic processes that have to come about. It's a whole new way of life. So we're dealing with the origin of new bodies, new ecosystems. It links geology, it links biology. It seemed like a wonderful problem to hang my shingle. And that's what I've been focusing on literally ever since I saw this diagram. Now what we're going to talk about today is paleontology and molecular biology. Now I began, when I saw that diagram, training to be a paleontologist, a fossil hunter. And we're going to talk about that. I'm going to take you on a field expedition with me that happened over a series, about a decade or so. And you're going to see how we go about finding new fossils that tell us about the great transitions in the history of life. But interestingly, as I launched those expeditions, there was a revolution happening in biology, a revolution in molecular biology. And that revolution so greatly affected me, as you'll see, there were a couple papers that a graduate student gave me that changed my career. So essentially I had to retool to some extent, relearn mid-career a whole new discipline, which is DNA technology, molecular biology, and developmental genetics. Yet both these approaches are in the service of understanding the great question in the history of life, which is how these great transitions happen. So we're going to begin with fossils, then we're going to switch to molecular biology to the other trajectory of my research. Okay, now let's put on our paleontological hats and let's begin thinking about how I was looking at this diagram from the mid or late 1980s. Now if you're a paleontologist and if you want to make headway into this problem, understanding how fish evolved to walk on land, you're going to go out and try to find new fossils. Now to do that, what you do is, the world's a big place. The places where we find fossils are very small. So how do you winnow it down to the places where we find fossils? Well, to do that, we look for places in the world that have three things. You look for places in the world that have rocks of the right age to answer whatever question interests you, right? So if you're interested in the origin of humans, you're going to go to rocks about 3 million years old or so, or you're younger. If you're interested in this, you're going to go to a time period called the late Devonian, based on other fossil finds. We kind of figure out that about 390 to 365 million years ago, which is when these fossils were found, is the window of time to be in. So first, look for rocks of the right age. Then look for rocks of the right type. Not every kind of rock holds fossils, for a lot of reasons. Maybe the critters didn't live in the environments that those rocks formed in. Or maybe those rocks were so deformed by millions of years of geological history that whatever is inside was destroyed. So you have to learn geology to learn what the rocks of the right type are. Finally, it does me no good if these wonderful rocks of the right type and the right age are buried 12 miles underground. Right? They have to be exposed at the surface. We don't randomly dig. So that's it. We look for places in the world that have rocks of the right age to answer our questions, rocks that have the potential to hold fossils, and rocks that are exposed at the surface. Now, the major breakthrough for us, ingredient X, our special sauce, was realizing this. That is, if we were to make progress in finding an intermediate, let me just go back a bit. If we were to make progress in finding, say, a flat-headed fish with arm bones inside of their fins, we wanted the right environment. And this was what we made the bet on in the early 1990s. That is, we would look for rocks of late Devonian age, around 380, 375 million years old, that reflected these environments, ancient delta systems, kind of like the Amazon today. Now think about that. The ancestors of creatures that are walking on land, they're not going to be in the middle of the ocean. Right? Where they're going to be is they're going to be in nearshore environments, in estuaries, in rivers and lakes and so forth. And it became very clear to us that if we can find places in the world of the right age that had these environments, a delta system embedded in the rocks in the ancient world, we would maximize our odds. And in fact, we knew if we found this kind of thing, we're going to sit on it and work it really hard and do all the hard work to try to find fossils inside. The good news is geologists can recognize these kinds of rocks that are reflective of these environments. Right? We know from sandstones and siltstones and shales, you can identify places in the world that had regions that were formed in ancient delta systems. No problem. All right, let's give you a sense. So we had this idea, right? So age, type. Now we ran around the world looking at geological maps, looking at the oil and gas literature, looking at geological literature to see where these rocks are exposed at the surface. Where can we dig? And I'll just give you an example. We had maps from South America, from Asia, you name it. Here's North America. And you can see here's a map of North America. And superimposed on that map are where the Devonian age rocks, again rocks in this case about 365 to about 380 million years old, are exposed. And they identify 3 areas shown in red that had the sandstones and siltstones and shales that were reflective of their formation in ancient delta systems. Okay, so there's one here in Pennsylvania where the arrow is. I knew about that one. That was, I had actually worked there early part of my career. There's a place in Greenland that Danes and Swedes have worked over the years that produced some early limbed animals. But now I think you see where we're going. Extending 1,500 kilometers east to west across the Canadian Arctic was a very special area. This is an area that had rocks formed in ancient delta systems, but these were older than the other 2 places. These were the right age, almost the exact right age when we looked at other finds. So that was it. So that's what led us to the Arctic back in the mid-1990s. To give you a sense of what we're doing is what you're seeing here is a map of the Canadian Arctic. In the upper left, you see a map of Nunavut territory where the flag of Nunavut is. And what's circled in red there is the whole territory. It's vast. It's, at its most northern point, it's about 400 miles from the North Pole. And if you look at the main body of the slide, what you see are the Arctic islands of Nunavut. These are a series of islands that extend east to west over about 700 kilometers. And circled in red here are where rocks of the right age and rocks of the right type are exposed to the surface. This was the map that we uncovered from the oil and gas literature, from the Canadian Journal of Petroleum Geologists, that really gave us the insights to lead our expeditions. Okay, so now we're presented with this wonderful area that's vast. So how do you crack into it? Well, you got to start somewhere. So we began in 1999 in the western part of the Canadian Arctic. And we didn't know anything about working there. This is our learning experience. And it's safe to say that this first year was a complete bust scientifically. We found nothing to take home. I found nothing that would ever make it into a scientific paper. I came home, okay, what are we going to do? But I learned and the crew and I learned so many lessons that year about how to work here. We learned, number one, we were in the wrong area in the western part of the Arctic. We were in the middle of the ocean. We needed to be in the streams and rivers, and I'll show you in a second. We learned how to camp up there. We learned how to avoid polar bears up there. We learned how to identify fossil bone from rock, which is essential, and you'll see how important that is in a few minutes. Okay, so we licked our wounds. Learned our lessons, though. It was a useful failure. So let's just look at this diagram. We were in the middle of the sea, so we had to move upstream. So geologically, what that meant is, following the geological papers, that meant moving further east. And this is in 2000 where we decided to go next, and this was a lot better. Well, you can see in the distance here, see those cliffs and mountains? Those are not terribly horrible. You can scamper up and down. Scamper is probably the wrong word. You can go up and down them. Those, if you were to look at those rocks, they were formed in ancient streams and lakes, perfect environments. The problem is, we started to find fish, getting closer, but these were fragments of fish, not whole skeletons. Here's the problem. Those streams were very, very fast moving. They contained giant boulders. It was clear from the geological record that these streams and rivers were too big. We needed a place in the Canadian Arctic that had very slow moving streams. And we needed to find the geological signature of what that would be. That's what we did. We went back a couple more times, and this is again a long saga in about 6 slides. We found this valley. Here you can see the valley, and you can see the rocks, they're sort of tilted, and you can see them in the distance, all the way extending to that snow field. We ended up finding in this spot a layer of fossils that contained hundreds of fossil skeletons of fish piled one on top of the other. We really hit the jackpot in terms of the number of skeletons. So we really, by this point, by about 4 or 5 years in, we had found a layer. But when we worked that layer, we had to work it for 2 more years, which in this case is summers, 2 more summers, because we weren't finding anything new. So we just worked that hard, hit it hard, worked it hard, until in 2004, this is the slide of the major discovery. Remember, by this point, we had already trained our eyes to see the difference between rock and bone. So those years where we weren't finding a lot of fossils weren't failures because we had the pattern recognition in our brains already. But the moment I saw this, and you can see in the tip here, you see a little V, that's an upside down jaw of a fish, and not just any fish, it's an upside down jaw of a flat-headed fish. Now you go, where is this? And you think I'm crazy, I know you do, but it's right there where that arrow is. You can't use color to tell it apart. You have to use shape. What you'll see is the shape of the rocks. They break with sharp angles. In the front area, what you see is a V formed of rounded structures that have the impressions of fish bones, if I was to show it to you. It was clearly a fish jaw. So we removed this and brought it back, and in that year we found 3 more of these. So we found 4 of these fish that year. We've since found 20. Bring it back in the lab, and this is in 2004. And we removed the rock grain by grain, and now you can see that what we have is the top of a head of a flat-headed fish. You can see one eye hole and another eye hole, so orbits. That took 5 months to prepare, by the way, by a preparator. Another 5 or 6 months go by, boom, look at this. We have a flat head with eyes on top. You can almost see when you go back there, you see sort of shoulder blades, and then you see sort of a neck area. Fish don't have necks. What's the deal? Okay, look, this is what motivated it. We were looking for a flat-headed fish with fins, with arm bones inside. We went to a place in the world that had rocks of the right age, rocks of the right type, rocks that were exposed to the surface, and what did we find? That, Tiktaalik roseae, which is a fish with arm bones inside. If I was to hold this in front of you, it's about 4 feet long, one of the smaller specimens. The biggest ones are about 8 feet long. It would look like a fish to you with scales in its back and fins, as you can see in the front right, foreground right, has fins with fin webbing. And there's lots of other fish bones in there as well. But if you look at the other things, if you look at the shape of the head, in fact, other features of the head, if you look at the neck, those are characteristics of animals that live on land. And it got even more interesting. When we cracked open the fin, what do we find? We found bones that correspond to the upper arm, forearm, even parts of the wrist of limbed animals. So it's really remarkable. And so when we were able to put this in a phylogenetic or evolutionary framework, and this is an old diagram because some new species have been discovered ever since, but what you see is the trajectory going from the base of this tree all the way towards the top. At the top, what you have are limbed animals. These are animals that are capable of walking on land. What you'll see, Tiktaalik sorts out as a real intermediate there, and you can see gradual changes in the structure of the skull as well as in other parts of the body. What's interesting is ever since we discovered Tiktaalik, it's not rare. There are 20 individuals that we now know. Other teams working in Quebec, Eastern Europe, and Russia have found species like it. So this is a part of a tree that we've gained increasing knowledge over the past decade and a half. Now notice how I showed you that what we did with the original discovery of Tiktaalik is somebody had to sit under a microscope with a fossil and remove the rocks grain by grain. In the last 15, 20 years, since the discovery of that beast, we now have high-energy CT scanners and advanced light sources, which can scan inside the rock to see the bone inside. In the basement of Culver Hall, that's where my lab is, which is to the west side of Botany Pond, there's a CT scanner, high-energy CT scanner, and we can put these animals in there. So this is, at the top, is that one specimen I showed you that has been put under the CT scanner. We can now see inside the rocks to see the vertebral column. We can see the fins. We can now see the internal structures in great detail. We can look at the fin itself in a number of different specimens. And we can see the upper arm, forearm, even parts of our wrist. We can see in blue the fin webbing, how it has both bones inside there. And we can even begin to map how the arteries go through the bones themselves, what the vasculature looks like, and map what the muscles would be and how this animal would move. We can also look at the joints inside the skull to begin to see how the bones likely moved as the creature fed. And we can constrain that by looking at the surfaces of what are called the sutures, or the joints of the head bones. And we can also look to see what happens in living fish as they acquire prey with these same joints. And it gives us an image of a living, breathing, feeding creature. And that technology did not exist in the same way when we discovered Tiktaalik. Anyway, so what we were able to discover is a creature that has both lungs and gills, that has fins with arm bones inside, that is part fish, part land living animal. But what it shows are two very important points. The first is that many of the organs and many of the inventions that were essential for animals to be able to walk on land already appeared in fish living in aquatic ecosystems. Wrists, lungs, necks, all this stuff was already in fish. So the inventions necessary for the major transformation happened well before, are very primitive. The other big piece, and this is what I did a riff on with my book and TV series, You're in a Fish, is really some of these events that we see happening in the Devonian in fish like Tiktaalik roseae and its evolutionary cousins are things that are in our own bodies. This neck we see for the first time in Tiktaalik and its cousins is something, when we trace the fossil record, that is our own neck. This wrist we see for the first time in Tiktaalik and its evolutionary cousins living in the Devonian is something, when you trace the fossils, becomes our own wrist. So every time you bend your wrist and every time you shake your head, you can thank these fish evolving to walk on land in ancient Devonian ecosystems. So if you look at our appendages, so if you look at a human arm or leg, there's a sort of certain defined pattern that's seen in all land-living animals with bones, vertebrates. There's one bone, two bones, wrist bones, and digits. And that same pattern is in the legs. And I've color-coded it there, and they're the scientific names for, they call those different regions, stylopod, zugapod, and autopod. Now we can trace that. We see that same pattern in bird wings, whale flippers, mouse limbs, frog limbs. That's true with every vertebrate organism. It has the same pattern. We can trace that all the way back to Devonian tetrapods, limbed animals, living on land 365 million years ago, and you can see the one bone, two bone, digit pattern. But now with Tiktaalik and its other cousins, we can trace it even further back, one bone, two bones, and then the terminal end. We can trace it back to animals with fins. What you see when you look at Tiktaalik is it has all three regions, but it doesn't clearly have fingers, and it has, you look at the terminal end of it, it has fin webbing, fin rays. So there are two things that are happening between Tiktaalik and tetrapods. The first is, and limbed animals, the first is the loss of the fin webbing, right? You could see that. And the other is the gain of a true digital arcade, right? So you have a loss and a gain. Now I want you to remember that, because we're going to talk about development next. And when we talk about development, that's exactly the issue we're going to be looking at, okay? So that's what fossils show us. It shows us the deep antiquity of elements of our own body already in fish. But it sets up other puzzles. How do you lose the fin webbing and gain digits? All right, so now we're going to talk about a change, some discoveries in biology that really changed the way I approach science. And believe it or not, those discoveries happened in some of the most unlikely place. They happened in fruit flies, Drosophila melanogaster. It's safe to say that discoveries in Drosophila melanogaster, the fruit fly, changed my trajectory of my career from being a fish paleontologist just straight up to somebody who does molecular biology. How could discoveries on a fruit fly, tiny little fruit fly, do that? Well, it begins with the work of T.H. Morgan, one of the great scientists of the 20th century. Morgan was interested in a lot of things through his career. Regeneration, when he worked at the Marine Biological Laboratory at Woods Hole. But then he shifted to genetics. And talking to an entomologist at Berkeley and a geneticist at Harvard, he realized that if he was to make progress in understanding genetics, he needed to work on an organism, a creature, that breeds quickly, is very small, you can look at a lot of mutants, and you can have a lot of them in your lab. And its life cycle is very short. You don't want to do elephants, use elephants to understand genetics. Fruit flies are much better. So what he did is he devoted a career to studying fruit flies, first at Columbia and later Caltech. And he, in one of the sort of most iconic places in all of science, he set up first at Columbia in Schermerhorn Hall and later in Caltech, he set up the fly room. And in the fly room, he had a cadre of incredible scientists, some of whom were going to win the Nobel Prize, like T.H. Morgan, in their own right later on. One of the geniuses in his lab was this person in this picture here, Calvin Bridges. Bridges was a remarkable person who knew how to work with fruit flies and developed many techniques to handle them. But he also had an eye for mutants and an eye for understanding how mutants can tell about how genetics works. So he was on the hunt for mutants, and he found some mutants that really changed the way a lot of us think about science. And this was in the 1920s and 30s. I didn't know about this at the time, okay? I was studying to crack rocks. I had no idea this stuff. It was only happened a little bit later. He discovered a mutant that was called bithorax. So what you see here on the left is a normal fly, and you can see it has one set of wings and one thorax. But in bithorax, as the name implies, he found a mutant that has an extra body segment, an extra thorax, two sets of wings instead of one. Now that's a big deal. Right? That's a duplication of a whole body part. That's something that's addressing the basic architecture of bodies. So whatever these mutants are doing, these are genes that are affecting the basic architecture of bodies. Now there's another mutant called Antennapedia. Think about that for a second, what that means. So here's the front end of a fly head, normal fly head, and what you're seeing are the eyes on either side, and there's like little tiny antenna there. What's Antennapedia? Pedia meaning leg. It has a pair of legs sticking out where its antennae should be. Antennapedia. These had legs sticking out of their head. Now, what Bridges and others were doing with these mutants is they're identifying genes that are building the basic architecture of bodies. That's what these genes are, telling us what organ forms in what place, giving the body that instruction during development. So these mutants were there for a while, and then this person, this great scientist, Ed Lewis, came by. And Ed Lewis was at Caltech, and he was a classical geneticist using updated versions of some of the techniques that T.H. Morgan and Calvin Bridges and others did in the fly room at Caltech. And he realized that these two mutants, Antennapedia and bithorax, they're actually caused by a number of genes, and they lie together on the chromosome. Okay, so what you have here is a whole bunch of genes that control the basic architecture of the body lie together next to each other in the genome. And interestingly, he did this, Lewis, without much molecular biology. He did it by looking at flies and crossing them with breeding them and so forth. It was really remarkable. All right, this all happened, I knew nothing about it because I was studying geology, I was studying paleontology. I came back from a field expedition one year in the late 1980s, and a graduate student, a molecular biology friend, came by with a stack of papers that he wanted me to see. And these are the papers. They're worth drilling into. This first paper is a conserved DNA sequence and homeotic genes of the Drosophila, Antennapedia, and bithorax complexes. You notice the names. Another paper is the same. What's the translation of this? They sequenced the DNA of these genes. That's a big deal. We now knew the sequence of DNA of the genes that are building a fly's body. But here is the kicker that caused the paleontology to put his geology books away for a little while and start picking up the manual for molecular biology, is the next paper said it showed that these genes were conserved in other metazoans. Translation, versions of these genes are in all animals. So the genes that build the basic architecture of flies, telling legs where to develop and wings where to develop and so forth, are present in frogs, mice, people, and yes, even fish. This is really remarkable. Let's drill into it because that's where it's going to lead off with our work. So here's a fly embryo. And I color-coded the fly embryo to show where these different genes are turned on during development of the embryo. So what you're seeing is the fly embryo, color-coded, and each of those different colors shows where a different gene is turned on along the body. Those genes are related to Antennapedia and bithorax, and they tell which segment of the body where to develop a leg, where to develop an antenna, where to develop a wing, and so on. Well, that's cool. That's cool about flies, and it's great. But what's interesting is humans and mice and fish have these genes too, versions of these genes, which are turned on in the body axis, and they tell the body during development which structures to form in different places. With these discoveries and others in other genes, I'm just giving you one gene family of these genes, they're called Hox genes, if you want to know, we're seeing an ancient genetic bodybuilding toolkit. That is, there is a toolkit to build animal bodies as different as flies and people and worms, that the versions of the same genes are active in these creatures, building bodies and building their diversity. You could see why I was excited. Right now, it's one thing to compare adult species, an adult Tiktaalik to an adult limbed animal, but now we can compare the toolkit that builds bodies. It's like comparing the recipe to make something, the recipe to make a cake, to the cakes themselves. By comparing the recipe, you're getting down to the mechanism by which they're built and how they might have evolved. This was a truly remarkable moment for our field, not only for me personally, but for our field. This was a moment where there was a great excitement about multiple disciplines coming together. And one of the great moments in our field happened here at the University of Chicago in 1994. I dug this old slide out. The 4th International Congress of Vertebrate Morphology, which is happens every couple years. It's a meeting where anatomists and paleontologists would get together and they'd talk about anatomy and how it evolved and how it works and so forth. But in the 1994 meeting, the molecular biologists came. And they came with those pictures that I showed you of flies and people showing the common genetic toolkit that builds different bodies. There was palpable excitement in the room where paleontologists, anatomists, developmental geneticists, straight up evolutionary geneticists, all saw a shared future with one another. And it was the birth of a sub-discipline in my field called, now known as EVO-DEVO, or evolutionary developmental biology, which is a multidisciplinary field. And then one of the origin stories for it, there were several meetings, but one of the most important in my particular branch of biology was in Chicago in 1994 in the newly opened Biological Sciences Learning Center. Now let's get back to this diagram because it really says a lot. Now these are the basic tools to build bodies, but what's interesting about them is they're also co-opted and repurposed to make different organs. What you're seeing here is, these genes turned on along the body axis, right? From front to back along the body, telling organs where to develop. But they're also recruited and co-opted to make other organs. These same genes are turned on in limbs to make arms and legs. These same genes are turned on again in the same pattern to make external genitalia. So different organs use similar sets of genes. That is, they're co-opted to make new organs. It's like a computer program with subroutines. These subroutines, the main program was evolved to make the body axis, but subroutines are making different organs. So if I look at limbs, what you have is these same genes that are making the body axis, some of them that we're talking about here are turned on to make limbs, to make different regions of the limbs, to make upper arm, forearm, even part of the wrist. So that's very powerful because that is the entry point for some of our own work. And let me give you an example. This is a mouse limb. And this is a mouse limb where the researchers, not my lab, where the researchers have mapped where cells that had one of these genes end up in the limb. And check it out. So here you have, in the embryo, genes were turned on, and you had asked the question, where did those cells end up in the adult? They were able to trace these cells throughout development. What you see in green is where the cells had the activity of this gene. And look what it does. It marks the wrists and digit area. It marks the hand. There's a little area with a tendon in there, but that probably is more artifact. It marks the wrist and digit area. So this particular gene is a marker for the hand and wrist. Now, what happens, here's another experiment to show you how important they are. What happens if you remove this gene from development? So genetic engineering had gotten to the point, this actually is an experiment that was done about 20 years ago. Genetic engineering had gotten to the point where you can actually make a mouse that's lacking these genes. So if you're lacking this gene, what would you be missing? You'd be missing the hand and wrist, and that's exactly what you see. On top is a normal mouse. The cartilages are stained blue, and on the bottom is a mouse that has these genes deleted. And what do you see? It's missing, it has one bone, two bones, but it's missing the wrists and digits. So these genes, which were active in making the body axis, now, in this case, are marking the terminal end of the limb, the wrists and digits, right? It marks them, but also when you remove them, they're necessary for the development of the wrists and digits. So the question is, what's happening in fish? Fish has these genes too. Let's see. Okay, so if we do this same experiment in fish, what do we find? This is what my lab, this is what Tetsuya Nakamura and Andrew Garricky in my lab did a few years back. Boom. We see that the equivalent of the wrist and digits in fish are the fin rays, the fin webbing. The wrist and digits in mice are the fin webbing in fish, the equivalent there. What happens when we knock these genes out, when we do that same experiment? When we do the removal experiment, just like you see in the mouse on the left, on the right what you see is when we remove these genes, the normal fin is up top in the upper right. On the bottom is we remove the fin rays, fin webbing. So what's the comparison that these genes allow us to say? They basically say that if you're comparing fish fins to human arms, that the cells that make the fin webbing in fish are equivalent to the cells that make the fingers and toes in limbed animals. And now the question begins to understand how those cells in these different species follow these different fates. Remember I showed you in the fossils that there was a trade-off between the fin webbing and the digits when we compare Tiktaalik to other creatures. Now we're seeing one plausible mechanism for that happening is by changes to these cells that are equivalent between wrists and digits and the fin webbing. So I was writing my most recent trade book, Some Assembly Required, which sort of talks about a number of these things. And I was finishing it up, and I was taking a break from the writing. I was reading an autobiography of Lillian Hellman. And we're trying to understand what does Lillian Hellman have to do with Drosophila genetics and fish fossils and so forth. She actually has a quote which is really relevant. She was looking at her own life, and her own life was a very storied one. I mean, just look at the picture. She was blacklisted by the House Un-American Activities, the McCarthy Committee. She was quite a creative and a colorful personality. But when she was looking at her own life, she came up with this quote which says, "Nothing, of course, ever begins when you think it does." And I remember reading that when she was thinking about Lillian Hellman's life. I was thinking, that's evolution and great evolutionary transformations in a nutshell. Why? Why? Because when you think about the origin of our hands, the genes that are so important for the development of our hands and feet originally arose patterning the bodies of things like flies and worms. The genes that make our eyes were doing other things in other creatures. They were co-opted and repurposed. Taking another example, all these structures, virtually all the structures that are used by animals to invade land and fish, originally arose in fish living in aquatic ecosystems. I mean, if you think lungs evolved to help creatures live on land, and feathers evolved to help birds fly, you'd be in very good company. But nothing, of course, ever begins when you think it does. That's completely wrong, and we've known it for over a century. Lungs originally arose in fish living in aquatic ecosystems, eons before animals took their first steps on land. Feathers arose well before birds ever took flight. They arose in dinosaurs as thermoregulation or courtship displays. This is true with history in general. Nothing ever begins when you think it does. But this principle also has practical value. And for the last few slides, this is what I want to show before we go to question and answers. When we think of certain traits, we tend to think of them as unique. Okay, take this. We should hope we had an inner salamander because salamanders do so many amazing things. What you see here is an axolotl. It's a salamander. And on the left it's showing one of its remarkable abilities. On the left at the top, you see a salamander limb. Now, if you cut that limb, or if it's bit off by a fish or another salamander, what you'll go to is the second one. You'll have a stump left there. So if a salamander loses its appendage, it'll have a stump. And then look what happens over time. As you go, follow the slide, the limb completely regenerates. The muscles, the nerves, the bones, the arteries, they all come back with a very high fidelity pattern. That is amazing regenerative capacity. And they can do this as adults. And I'm just going to drill into a little detail here because it's actually relevant. Is if you look at a cross-section of these appendages as they develop, okay, as what happens as it regenerates. So here's the salamander. Look at the top, and what you see is the salamander appendage, the stump, okay, that was cut off. After 1 to 2 days, what you have is like what happens in us, wound healing. You have the closure of the wound. You have the formation of a vascular supply. You have what's called a wound healing epithelium. It's a particular kind of tissue, just like us. We stop there, okay? We stop at wound healing, if we're lucky. Then what happens, starting at 7 days, what you start to see is a cluster of cells begin to form at 7 days at the terminal end of that stump. And these cells are very, very special because what they're going to do is direct that regeneration. All right, so then you go to 1 to 3 weeks, and all of a sudden, cells in that cluster called the blastema that began at 7 days, they're going to start to tell the cells to form muscles and nerves and bones. Cells begin to differentiate into different types. And then at the bottom, a pattern forms, a new limb emerges. Okay, this is important because first, we stop at 1 to 2 days. We don't have wound healing. We have wound healing. We don't have regeneration. But then the key thing here is the formation of that cap of cells called the blastema. Now, why am I drilling into that? Well, if we look at regeneration, we can ask, what other creatures regenerate? Are salamanders the only ones that regenerate their appendages? Well, a former postdoc of mine, Nadia Freubisch, studied regeneration, but then she started to look at the fossil record. And she found in the fossil record, going back at least 300 million years, the telltale signature of limb regeneration in amphibians, in fossils. And these are from fossils that some of them are 300 million years, some are a little bit older. So it clearly goes back very deeply in amphibians. Okay, well, let's push it even deeper. What about fish? What happens to their appendages? Well, I had a postdoc in my lab, Randy Don, who had these fish in the upper right, a fish called Polypterus, and they're fish with a giant sort of fleshy lobe there. They happen to have beautiful fins, and they kind of walk in the water a little bit, but they also happen to be very mean, and they bite each other's appendages off a lot, which is bad for them, but it's great for science. Because what happened is Randy watched them, and here's what happened. This is a cut he did. It turns out they regenerate. Here you can see on the left, there's a cut limb. After 7 days, they have a wound healing epithelium. They heal the wound. After 14 days, they regenerate. And look at this. You can watch the pattern inside. You can stain the cartilages blue and the bone red. And look at this. After a month, you start to see a pattern. After a month and a half, that pattern emerges even better. After 3 months, there's bone. After 6 months, you compare it. There's the regenerative limb on the bottom. There's the wild type or untreated limb on the top. They regenerate. And guess what? If you look at more fish, they regenerate. If you look at the evolution of regeneration of appendages, fins or limbs, look at here's a phylogenetic or evolutionary tree. On the top are amniotes. Amniotes are you and me and birds and reptiles, so that branch of the tree of life. But pretty much everything from amphibians to amphibians' closest cousins, lungfish, to the cousins there, polypterids, paddlefish, gar, and other fish. What we see is in blue, they all regenerate. Regeneration is not something that's unique to axolotls, to ambystoma, that salamander I showed you. It's primitive. We lost it. So that changes asking the question of what happened in our history that caused us to lose it. So 3 postdocs from my lab got together and compared the regenerative mechanisms in different fish and amphibians. And what they did is they looked at the regenerative appendages, they cut the appendages and they saw them regenerate. And then they used techniques where they can separate the cells and identify every single gene that's active in that patch of cells. Remember the blastema, that patch of cells? Every single gene. And they compared them. And what did they do? They found a common regenerative specific regenerative program. That is, they found a common set of genes underlying regeneration in all these different creatures, which we lost that activity. So the question is, how can you re-stimulate that activity? How do those genes act? We have the parts list now. We know which genes are likely involved in regeneration. But now we have to do the hard work. This is the hard work in biology, is understanding how these factors interact to produce to produce new structures. But here's a case where regeneration, nothing ever begins when you think it does. It's far deeper. But that's an advantage for science, because now we can look at what's common among all regenerative animals that regenerate their appendages and compare it to creatures like us, which do not, and to ask the question is, how can we stimulate better wound healing or regeneration? So I hope I've had a chance to convince you that the linkage between paleontology and molecular biology can be a very powerful approach in the history of life, using embryos and genes and fossils. You can ask the question, you can ask, okay, well, who really cares about you and your fish? And that we have this history with fish. And I would say my answer to that would be the obvious one for biomedical research is if you look at the Nobel Prizes that have been awarded in medicine and physiology in the past 40 years, they've gone to people working on your inner corn, your inner fly, your inner mouse. They've gone to people working on mice, flies, corn, yeast. 2 Nobel Prizes awarded to 5 people in the last 15 years have gone to people working on Caenorhabditis elegans, which is a tiny little worm the size of a comma on a piece of paper. Yet that little worm is telling us how our genes are naturally silenced during growth and development, how our genes are programmed to die appropriately, and what goes wrong in diseases like cancer. I like to think that as we discover cures to everything that ails us, from Alzheimer's to different cancers, that the breakthroughs that will extend and enrich our lives will in some way be based on flies, worms, mice, and in some cases even fish. I can't imagine a more powerful or more beautiful statement on the importance of understanding our evolutionary connection to the rest of life on our planet than that. Thank you very much. Thank you. I wouldn't be standing out here today if it wasn't for scores of people. Literally 50 people have helped me in the field. I did not discover Tiktaalik alone. I've led expeditions with Ted Deschler and the late Ferris Jenkins from Harvard University. I've had support of the Inuit and indigenous communities from the north. They've supported us in so many ways. I've had financial support from private foundations, the Princeton Foundation, from the Biological Sciences Division of the University of Chicago, which has supported me very generously, NSF, National Geographic Society, and so forth. I've listed members of my current lab who've had to tolerate long absences to write books and do field work and so forth. And then I've got to single out my friends Cliff Tabin and Sean Carroll. Cliff is a head of genetics at Harvard University Medical School, Sean Carroll at Howard Hughes Medical Institute. Both of them, when I saw those flies papers, they were friends of mine. I saw the fly papers, I called them up and I said, help, how do I become a molecular biologist? And they helped me. And finally, there is kind of nothing that happens, whether you work in Antarctica or the Arctic, without family. So thank you, Michelle, Nathaniel, and Hannah. Thank you.
So we'd like to take the opportunity, if anyone in the audience has a question for Neil, I can happily bring you the microphone. Any questions? Thank you, Neil. That was wonderful. Lots to think about, I know. Oh, okay, wonderful. I'll bring that down.
Thank you, Neil. That was great, as always. And it was great to hear you jump into regeneration at the end, having heard you last week on the subject of regeneration. Richard Gray, great friend of the university, art dealer, in talking about art with art historians, said you people have great hindsight. You can tell us what happened. Collectors have insight as to what particularly appeals to them. But I, Richard, speaking as a dealer, need to have foresight. Now, just at the end, you mentioned Alzheimer's and cancer. Jump 10 years in the future, if you would, and tell us what's going to happen with the foresight that you have.
Okay. Well, the first is I work in Antarctica, okay? So the one thing about Antarctica is climate change, what you're seeing here is Antarctica actually, and this is the lead slide. Climate change is very real. So we have to be worried about changes in sea level, changes in the climate that my own children and your grandchildren are being raised in. And it's a very real issue, and it's very hard to escape when you work in polar regions. It's not hypothetical at all, particularly in the Arctic where we work, it's been opened up. But when you think about the power to manipulate cells, when you think about the power to manipulate genomes, when you think about the computational power we have in terms of, so right now in biology, we're very good at generating parts lists. I can tell you every gene that's active in particular tissues and development, but less is known about the networks and genes and how they come together to build bodies. But I think we'll be able to take wound healing a little further to have if the regenerative work works out in many ways, people will be able to heal without scar tissue in many different cases. I don't think we'll see regenerative limbs, but I think scar tissue will be something that will be greatly minimized for particular injuries. I think genetic technology has gotten to the point where it affects everything we do. It affects everything from agriculture to human health to even fields that I don't even know, they can't even imagine yet, because we can manipulate pretty much every aspect of organisms. And that what sets up not only for health and disease, but also in terms of how we interact with the environment. When you think of biotechnology and genetically modified organisms, and what we can do with technology to expand our own senses. No, it's one, when I think about the world when I first started molecular biology, it was an event when somebody sequenced a gene. Then it was an event when somebody, a team, sequenced a genome. And now we're seeing genomes, loose genome, not rough drafts of genomes sequenced in an afternoon, for a small amount of money. So that amount of technology at our fingertips, including imaging technology and computational technology, changes the way we see living things. I think we will, if I had to bet, that we will find evidence of life on Mars at some point in the distant past, certainly water. I think, when you think of scientific discovery and our ability to see the cosmos and our ability to manipulate genomes, we're in a golden age of science. And it's hard, when you look at life, sometimes you get pessimistic about certain things, particularly during the age of COVID. Boy, but when you think of scientific discovery, you can't help but be optimistic. So that's my answer.
Excellent. Well, I'll give you some time to think. We have a question from the live audience from Rodolfo Probst. Fantastic talk. I was wondering if you could talk a little bit about how CRISPR/Cas9 has been impacting the field of evolutionary biology and how evolution apparently has been more thrifty than novel.
Yeah, so in terms of CRISPR-Cas genome editing, which is what I was talking to John McCarter about, we use it in my own lab. I mean, it's something, the Doudna-Charpentier Nobel Prize, which was this year, their paper came out in 2013. Within about a year or two, everybody was using CRISPR-Cas because it's a genome editing, because it's very effective, it's not very expensive, and it works in a lot of different species. So we can now manipulate the genes of a diversity of species and compare them. There's some people who are looking to see not only what happens when you knock out genes, but what happens if you make a fish gene much more like an amphibian? What happens if, does the fin change in some ways? So it gives us a new means to make genetic comparisons. We can also begin to see how genes interact with one another and how they work in networks, because we can remove a gene to see what happens to other genes and their activity and begin to see whole gene networks in evolution. So no, it's been a game-changing technology for us in the evolutionary biology. Now, in terms of evolution as thrifty, that's true. When you think of evolution, it's, when you think about how major changes to genetics and development happens, it's usually through tinkering. It's usually, as the Nobel laureate Francois Jacob said, it's through duplication, copying genes and diverging them. It's through co-opting genes, using them in new settings, like I showed you with the genes that build the bodies that are later used in limbs. It's from repurposing things in different ways. So innovation happens by modifying what exists. So that's why when I use the Lillian Hellman quote, "Nothing ever begins when you think it does," it's basically that evolution's very thrifty. There are very few. When you think of it that way, then novelties become less novel. It's always modifying what already exists.
Excellent. We have another question from the virtual audience from P. Saras. What about the lateral line of fishes? Did it evolve into something else, or did it just disappear?
Oh, it's still there, and it's doing great. So there are a lot of fish with lateral lines, and the lateral line organs are very important for their very important for their sensory system. What you're thinking of is what's happening with the lateral lines in creatures that shift on land. Where the lateral line system is lost is when creatures leave the water. So when creatures leave the water, that's when the lateral line becomes useless. But interestingly, there's parallels between the internal bits of the lateral line organs and inner ears, of our own inner ears, so hair cells and things like that.
We have time for another question if anyone would like. Oh, down here. Perfect.
Wonderful talk. I'm curious of going backwards a little bit in the sense that how do we lose our ability to regenerate? Can you shed some light onto that process?
Well, you asked the million-dollar question, right? So why did we lose and there's a lot of speculation about that. One speculation is that the immune system is always cleaning out cells. And one argument is that maybe that having a more effective immune system, like in mammals and birds, is such that it's a trade-off, that you lose the ability to regenerate because you're not able to keep the kinds of cells that are necessary to do regeneration. So people have mapped that, sort of the kinds of immune responses that creatures have versus their regenerative capacity, and there's a relationship. So it may be that there's a trade-off in that. That's one. That's a speculation. The other is, when you think about some of these regenerative systems, for the most part, though not exclusively, they're seen in aquatic animals that live in water. And for an animal that lives on land, and let's say that animal, let's say one of our distant ancestors, like an Australopithecine in Africa 3.3 million years ago, if they lost a limb, they wouldn't have time to regenerate. 5 months, they wouldn't live 5 months. On a savanna plain, they'd be dead. Whereas in an aquatic system, a creature that loses a fin can still swim around because they can use their back and other fins as well. So the aquatic system seems to be much more forgiving. So in an environmental sense, much of this seems to relate being aquatic versus terrestrial. In a molecular sense, it may be a trade-off, and the speculation is maybe it's a trade-off between sort of immunity, kinds of immunity, and regeneration. There may be other mechanisms we don't know about, but that's the million-dollar question, of course.
We actually have two last ones here from the virtual audience. Good morning from Japan. I'm interested in how the ribs change through time from in water to on land. This is because the modern amphibian rib cage seems to be insufficient to carry their guts.
Yeah, so if you look at Tiktaalik roseae, the creature we found, it has incredibly large ribs. And I don't know if when I showed that CT scan of it spinning around, you could see big ribs on that. So the creatures at this phase of Tiktaalik and some of the early land-living animals had massive ribs, and we're thinking that they were actually part of the body support to support them. Ribs shift in function. So remember, ribs have multiple functions in animals. Ribs support the body, right, particularly in four-legged animals. They protect the organs inside, but they're also used, once you have lungs, for respiration, for breathing. So there's the shift there. So once an animal gains a kind of lung where expansions of the rib cage are necessary to bring oxygen in, that changes the functions of ribs as well. Then all of a sudden they have to become quite mobile. So remember, ribs have lots of functions, and we can trace them over time, from locomotion and support to respiration.
Great, and I think we have one last question here for you, Neil, from Felix D. Do we know of any comparable Devonian species to Tiktaalik that were transitioning onto land but didn't make it? Do these evolutionary dead ends exist, or was Tiktaalik a more unique occurrence?
No, Tiktaalik is part of a family, a radiation of creatures. So we now know there are creatures. There was a beautiful one described last year from Quebec. That's Tiktaalik-like. There's another one from Eastern Europe that's very Tiktaalik-like. There's some more from Russia. So there's a whole bunch of these creatures out there. In fact, if you were to look at creatures evolving in the late Devonian, there are a number of creatures that are living in this sort of transitional zone between water and land. So it's not just one and done. It's not just one species. It's many.
All right, I lied. There is one last one that came in. This will be our final question. Mary Yurko says, why is the head to tail of the body represented on the genome from head to tail along the chromosome?
It's a good question. So that's for the Hox genes, for these genes I'm talking about, that was one of the most exciting things, that there is a structure to the position of these genes and how they're active in the, not only where they're active in the body, but when they're active in the body. And there's a whole explanation, it's called collinearity, and there's a whole lot of explanation for that. In the explanation lies, and much of it, in the way these genes are regulated. And by regulation, I mean the mechanisms that turn these genes on and off, and the switches that control them. So it's a whole apparatus that works together to turn these genes on and off, and it depends on where they sit in the genome, and then where the switches that control their activity sit as well. So, and it's higher order. So when you think about genomes, it's not just the gene sequence, but it's the higher order structure of DNA that's important here, and that's part of what's guiding us.
So I hope that I'm Kenneth Polanski, Dean of the Biological Sciences Division, and I hope we can all agree that this was an absolutely wonderful occasion to recognize and celebrate scholarship and science at the highest level at the University of Chicago. And really thank you, Neil, not just for the quality of the work that you've done, but the clarity of the presentation. And I thought what was so notable is how you gave insights into how scientists think and how hypotheses are generated and how data is interpreted. So that was really terrific. I'd also like to just acknowledge Bob Zimmer in his introduction did mention it, what an important advisor in science Neil has been to the university, not just in the Biological Sciences Division, but to the provost. His role in integrating the Marine Biological Laboratory with the University of Chicago has been notable. So I'm absolutely delighted that you have been recognized in this manner, and congratulations. And now to wrap up the event, Alison Ranney is going to come to the podium, so we're delighted that you are here and able to do this. Thank you very much, Alison. Sorry, I didn't hear.
Alison Ranney, can you hear me? Yes. I actually am here with my brother Ben Ranney, and we're really just delighted to represent the Ryerson and the Ranney families. As Bob Zimmer mentioned, this is a really special night for our family every year, and especially this year after a gap of last year. What's really special for me, I think, this year is the fact that there are so many, not only Ranney and Ryerson family members around the country and the world, but all sorts of other people who are able to enjoy this tonight. That's really a silver lining here of everything that's going on. Especially, I'd like to just recognize our father, George A. Ranney Jr. He has just absolutely adored this university for his life. He's the third generation trustee from our family, and he's really taken very seriously the idea of carrying on his grandparents' tradition with what they started here with this lecture. And he's always been very clear with our whole family and with all others that it's not about our family, it's about celebrating the university, the scholarship, and their collective leadership. And we saw that tonight. Tonight, in my view, was a really, Professor Shubin, it was just an underscoring of the importance of science, especially right now in our world. The last year has really brought that home for so many of us. But thank you so much for just that wonderful talk about, that I could relate to with Evo Devo and talking about inner fish and salamanders. I really went there with you, and I think my great-grandparents would have loved to see you in your flow. There's something so joyous about a scholar who goes there the way you did tonight, and that was really, really special to see. I wanted to close tonight to just say a final thank you of the evening to Bob Zimmer, President Zimmer. Bob, you have been, since 2006, you have just been such a wonderful partner to our family. You have been patient and kind and gracious. Your entire extended team has been with our family. We appreciate that every year. You really have made us feel a part of the university and also helped us focus on the university instead of our family, but understood the tremendous significance of this evening to our family. So Bob, thank you so much, and I look forward to seeing many more of you here next year, Bob included, and we are really looking forward to continuing the tradition. Thanks so much.